Welcome to the first lecture of this season's Midsummer Nights Science. And we have, I guess, a little bit of a theme of immunity this year. And so that'll be nice, I think, for people who come to a few of them to essentially deepen each time. So what I wanted to tell you about today was how the immune system works and trying to capture some of the challenges and questions that we're dealing with on a daily level when we study the immune system. And if we have, if we have time, I'll also talk a little bit at the end about the methods that we use at the Broad Institute to approach some of these questions, more genomic and large-scale approaches and unbiased approaches, if we have time. But I really want to leave time for questions as well at the end. So I'm an investigator. I have a lab here at the Broad Institute and at the Massachusetts General Hospital across the river. And my lab studies the immune system in many different ways, but our main question is how the immune system senses different pathogens in the environment. So with all the pathogens around you, how do you know which one you have seen and how do you respond appropriately? That's the question we're asking. But today I'll give you a much more general talk about what the immune system is. So first of all, let's define the scope of the immune system. The immune system is a collection of cells and organs throughout the body. Unlike many other parts of the body, it is quite distributed. The lymph nodes and the spleen are where a lot of the activity occurs. That's where the T cells and B cells of your body live. And as you know, the lymph nodes are scattered throughout the body. You can feel them when you get an infection because they expand. The bone marrow and the bone throughout your body is the site where the immune system is born. All the cells of the immune system are born there. And then they travel through the blood and through the lymphatics, which are another parallel vascular system that you don't think about very often. And they travel throughout the body and go really to all tissues, looking for any events that might be interesting and something that they have to do something about. And as you'll see as we talk today, there are many things that they're involved in doing. The immune system is composed of many different kinds of cells, but macrophages, neutrophils, T cells, B cells, mast cells, those are the main ones we'll mention at least today. There are a few others, but those are the major ones. And they exist throughout the body. So the immune system, besides being in all these organs like the lymph nodes, spleen, thymus, bone marrow, is also in every single tissue. So there are immune cells in the skin, in the brain, in the kidney, in the liver, everywhere. And the reason you need that, obviously, is because you could get infected anywhere. So it's not good enough to have the immune system in some localized place. So the immune system is really distributed. Before I start really talking in detail, I wanted to have a little glossary to make some definitions to make sure everything is clear. The first thing is what a pathogen is. A pathogen is a microorganism like a virus, bacteria, fungus, or parasite that infects us and grows with our help. We help it grow and causes trouble. So pathogens cause trouble. Commensals, which are, for example, all the bacteria that are in your gut, many of which help you, are definitely not pathogens. They don't cause disease. They don't cause trouble. So pathogens are really the ones that are infectious and cause problems. The immune system I already defined as the collection of cells and organs whose job is primarily to eliminate pathogens, but there are also other things that the immune system does. A vaccine is usually an inactivated pathogen. That is, one of these pathogens up here killed, or parts of it, and those are injected into the skin or muscle or other places, and they activate a very strong immune response against that inactivated pathogen. So when the real pathogen infects your body, just by chance, somewhere in the future, your immune system remembers the response it had against that inactivated pathogen and is able to destroy it before you even know that you've been infected. So that's the definition of a vaccine. Two other words that are often confusing. Antibodies are proteins that are made by B cells of the immune system, and we'll talk about them. And they help destroy pathogens. They recognize pathogens, bind to them, surround them, and help destroy them. Antibiotics are unrelated to antibodies, and they're drugs that are made by pharmaceutical companies and made by some small microorganisms too, that kill bacteria typically, like penicillin. And again, those are not part of the body. But both of these are very critical, obviously, in eliminating infectious disease, both antibiotics and antibodies. So there's a whole large variety of pathogens out there. And these are just pictures of some of the bacteria and fungi and viruses and parasites that are out there. There are tens of thousands of different pathogens that we encounter during our life. And many of them don't cause any harm because we eliminate them fairly quickly, and we just feel a little bit of fever and other symptoms for a week. But some cause serious problems. And just to give you a sense for how many pathogens there are, viruses themselves, this is a list of all the viruses here, and this is just a close-up of one little part. There are 2,290 that are documented at this point, but there are probably more. And bacteria, I can't even show. This is just the family tree of the different arms of the family. There are different branches of the family, but there are many of them, as there are fungi and parasites. And worms as well. There should probably thousands of species of worms that infect people throughout the world. So that's a lot for the immune system to deal with. And that's really the challenge for the immune system, is to handle this incredible complexity. So viruses can infect, this is just viral infections, can infect every tissue of your body, and there are viruses that have specialized to infect almost any tissue. And some of these viruses cause symptoms in those tissues that are quite bad. And other times, we can take these viruses and use them as tools to insert genes or to manipulate different tissues. So many immunologists and other biologists use viruses for different purposes. So what are we really up against? Well, today's epidemic scorecard, that is right now, tuberculosis, malaria, AIDS, diarrheal diseases, a few others, are really the major infectious disease problems. This scorecard would have looked different 50 years ago, 100 years ago, 150, 200. As vaccines change and as antibiotics get developed, this scorecard changes. Tuberculosis infects a couple of billion people right now in the world. So that's a pretty large number of people. Malaria, about 500 million people are infected, and so forth. And these will kill a certain percentage of those people, depending on the conditions of, for example, nutrition or other health conditions, co-infections, for example. Now, the reason these are up here and are so large is because we actually haven't worked out vaccines to prevent them. And so that's why they remain up there. They're not necessarily any worse than any other disease we've seen, but for some strange reasons, we haven't been able to develop an effective vaccine for these large, for these major infections. Okay, so what we're going to deal with today is two questions. What is your immune system doing for you today? What does it do on a daily level? And what could it do for you tomorrow if we help a little bit? Okay, so those are really the two questions I wanted to address today to get you thinking about what's happening on a daily level and what we might be able to do. Okay, so what is the immune system doing for you today? So in a most general sense, it's checking for pathogens and mounting a response against them. So let's say this is the skin, and these are little bacteria that may live outside the skin, and if you pierce your skin, they start to come in. Here are some cells called dendritic cells and macrophages that can sense these pathogens, and they can tell which pathogen comes in. They migrate as quickly as they can down these lymphatics, which are vessels, to the lymph nodes. And again, there are lymph nodes throughout the body, so they go to the closest lymph node. And in the lymph node, they alert the cells that are living in the lymph node, the B cells and the T cells, what they saw appear. And now the T cells start to surround these cells and learn from them what was seen up there. They start to divide, and then these guys migrate back out to the tissues and try to eliminate the pathogen. So this is what happens on a daily level every time you encounter a pathogen. So I want to show you some movies of what the cells of the immune system actually look like live in action doing these things. So we'll start with a simple movie, which is a very famous and very attractive movie, showing this lovely looking neutrophil, which is one of the cells that's very good at killing bacteria. Okay? So it sends out all these toxins everywhere around it when it encounters a bacteria, or it eats up the bacteria and kills it inside. So one of the amazing things about these cells is they can recognize bacteria. So here's a movie that shows you how it looks when it recognizes the bacteria. So there's the bacteria, the little guys over here. And it's doing a pretty good job of tracking it. The microscope had to keep moving over just to follow it through the slide. There it goes. It looks alive, and it is. And gone. So I find this a very, this movie encapsulates a lot of what the immune system does all day. It's basically sensing these things and killing them. And so even when you get a non-pathogenic infection, when you get a cut, it's doing this. Because, of course, the immune system has to just eliminate the bacteria that come in, regardless of whether they're pathogens or not. OK, so this is what happens at a daily level.
Is that bacteria actually running away, moving away from it, or is it just...
No. The bacteria, they do have ability to, certainly, they have flagella, which allow them to, kind of like a paddle, move it back and move it around and move. But it's not noticing the neutrophil, as far as we know. There are some sensory capacities that bacteria have to notice the immune system, but it's not at this level of fineness, like that it can tell where the neutrophil is. OK, the second movie is actually not in a dish, but actually in a mouse. And one can actually image fluorescent cells in a mouse. So I want you to look at this circle here. In red are bacteria called Listeria. Okay, Listeria is a bacteria that is found in, for example, raw cheese and cold cuts and pate and vegetables. And it's the one that pregnant women need to avoid. It's really, the most risk is for the baby that's in utero. So don't eat those things when you're pregnant. And so Listeria is in red. There are little dots around the place. And here are different kinds of immune cells. These are actually macrophages mostly in green. And this is just a movie showing what happens. No one did anything. This is what the immune system does. So watch this little circle here where there are some bacteria in there. You see that all the T cells within minutes, and I'll show you again, simply come over from everywhere around and accumulate in there. So really, they're mounting the forces against it. So in the dish, you see that one cell moving around. In the body, you have thousands of cells coming in at once and surrounding pathogen, trying to eliminate it. But it's not enough to just use the cells that are sitting around in your body. It ends up that you need to make more of them. And so one of the beauties, of course, of life is that you can just take a cell and make more of them. You just need food for that. So if you look in the circle on the top left, this is again in the context of an infection. And this is happens to be a T cell. And if you see this cell here, it's getting bigger and bigger, and then suddenly splits into two. OK, so now you have two T cells where you once had one. And when you have an immune response, this cell division response is induced by the bacteria. And so what happens is the T cells start to divide. And every time they divide, you double the number. If you do that 10 times, right, it's 2 to the 10th. That's a 1,000-fold. That's not many divisions, 10 times. You got a 1,000-fold increase in the number of T cells. So that helps you fight the bacteria or the viruses very well. OK. Another thing that the immune system does is it remembers what it encountered. So here's an infection, let's call it the first time you've gotten infected with something. Here are there's the graph showing the number of T cells and antibodies from B cells that are being turned on after a week. And that eventually turns off as the infection disappears. You might get a reinfection right away and a response, and you won't even notice it because the response is so strong. But then months later or years later, you might, of course, encounter that same pathogen again. That memory allows the response to happen in a day rather than a week. Okay? And it happens even stronger in a day than it does in a week at the first time. So when you get infected the second time, you don't even know that you're infected. It eliminates so quickly. Sometimes you feel maybe a little dizzy at night or funny or weird, and then it disappears by the morning. Some of those things are probably mild infections that you don't even know about. And so it really remembers very well. A good example of that is on these islands, the Faroe Islands, which are between Greenland and Scotland, where they had a measles infection in 1781 that caused a lot of death because it hit almost the entire island. And then there was no measles for 65 years. 65 years later, there was an outbreak. And everyone who was infected in 1781 was not infected in 1846. So the ability of the immune system to keep a good response up is quite impressive, given that measles really infected everyone before and now infected only the people who didn't get it before, which of course was most of the people 65 years later. So you get long-lasting immune protection. But why is it that we get influenza pandemics then? So influenza, we see it every year, and yet again, the year after, or a few years later, or 10 years later, you suddenly get this pandemic where everyone's getting the influenza virus and very few people are protected. And the reason in that case, and this is not true for most pathogens, that influenza avoids the memory by changing its molecules. So it actually changes the sequence of the proteins on its surface and causes a pandemic because that memory is lost. And so now the population looks naive to the bug. And it can spread again as much as it did the first time. And so that's the cause of the pandemics. But for most pathogens, if we've seen them or been vaccinated against them, we're OK. Influenza, you have to get vaccinated every year because it changes. Another thing the immune system does for you is protect you from really nasty diseases like smallpox. Because smallpox, there was a campaign to eradicate it. And actually, Jenner, who had one of the first people to figure out how to make a reproducible vaccine for this, predicted a few hundred years ago that it would be eradicated. And it was eradicated in the late 70s. And so we don't even need to be vaccinated anymore for smallpox. So that's a pretty impressive idea if you think about it, that the Earth contained this virus, and then suddenly, by just systematic vaccination, it was eliminated from the Earth. And smallpox is not a good thing. You don't want it around at all.
The left side of the chart shows millions.
Yeah, no, at this point, 1965, it probably would have been, I don't know. I don't know what the scale is. It might have been millions. It certainly was earlier. I don't know if this is like in the middle of the vaccination campaign or not. I don't know the exact years. So what the immune system is doing for you, it's checking for pathogens, mounting a response. It's chasing actively and clearing a pathogen just to sit around. It's chasing them to get them. It's mobilizing all its forces around a pathogen. And it's expanding its forces by cell division. So it doesn't need to call in forces. Each little force can make 1,000 copies of itself, just like the Monkey King in mythology that expands itself by cloning. So, and most importantly, I think, we're protected from future infections by remembering both vaccines, which are man-made, and natural exposures. So this really encapsulates, I think, what the immune system does broadly. Okay, so what is the immune system not doing for you today? What are the problems that can occur? And I'll go over those briefly, and then I'm going to go in depth into a little bit more issues. One is autoimmune diseases. This is not something the immune system doesn't do for you. It's something that it does too well, where it attacks the body's organs. And so there are autoimmune diseases in every tissue, practically. Multiple sclerosis, nerve degeneration, psoriasis, where you get skin rashes, diabetes, where the pancreatic beta cells are destroyed by the immune system. Crohn's and inflammatory bowel disease, where colitis, where the epithelial cells of the gut are injured by the immune system. And rheumatoid arthritis, where the joint are destroyed, the bone and the cartilage in the joints are destroyed by the immune system. So these are places where the immune system is doing exactly what I showed you in the previous pictures, except that there may not be a pathogen there. Now, why it does that is a very difficult question, and in the last, I think, 5 to 10 years, there have certainly been some genetic insights as to what mutations in humans give rise to autoimmunity. But honestly, we don't know what initiates autoimmunity. I don't think there's a case of common autoimmune diseases where we can say this is what started that person's autoimmune disease. And so that's an important open question. But in general, this is a problem that occurs because you've had to deal with 10,000 pathogens over millions of years. You have to make a pretty strong immune system. And one of the risks of making a very effective immune system is to make mistakes, of course. So it's trigger happy. Another one is immunodeficiencies. This is just a list of some of the immunodeficiencies known. They have different names like DeGeorge's syndrome, SCID, X-linked agammaglobulinemia, Wiskott-Aldrich syndrome. And these are ones that have different defects, like some people who have no T cells or B cells, defective production of antibodies, loss of macrophage function, and they all have consequences. Some are general, meaning you can get infected by anything. Others, specific kinds of bacteria or viruses, you become more susceptible to. And so these are called primary immunodeficiencies. These are genetic diseases. They're not common at all, but they're seen in children's hospitals primarily because infants usually come in with recurrent infections, and then they try to figure out what's going on. But the only solution to any of these traditionally has been bone marrow transplant, so getting an immune system from a matched donor that you won't reject. People are also interested in working on gene therapy approaches to solve some of these, and that will take a long time, I think, till it works well enough to be used routinely. Okay, the next problem that we have is this, and this is pathetic. I mean, this is a flower. It's a beautiful, fragrant thing, and here's this poor guy sneezing to it. And of course, it's not such a big deal to be sneezing to flowers, but it is a big deal when you're allergic to foods, as we've seen in the Western countries in the last 10 to 20 years, this increase in food allergy among children. And those can get quite dangerous. And there are many allergies that are dangerous across the board. And I'll come back to allergies in a minute. And then finally, there's also an appreciation now that the immune system, or inflammation, which is the activation of the immune system, can contribute to heart disease, cancer, Alzheimer's, and many other diseases. Now, why is that? Why would it have anything to do with those diseases? And the answer is very simple, which is that the immune system deals with any injury in the body. Anytime there's any wound or any injury or anything that causes any damage anywhere, it doesn't matter what the cause was originally of it, the immune system has to come in. It has to come in for two reasons. One is it cleans up the wound, just like you have to clean a wound before you put a bandage on it. The immune system cleans up all the debris and all the dead cells that are in the wound after the wound occurs. It cleans up, of course, any bacteria or other things that may have come in. But it cleans it up, and then when it's done cleaning it up, it tells the cells of that tissue or organ to start remaking the tissue. Okay? So those can come in, divide, and seal it back up. But the immune system has to come in and clean it up first. So wherever there's injury, to whatever organ, so even a cancer you can think of as an injury, the cells are growing out of control in an area, and they actually cause an injury to the surrounding area. These cells are not supposed to be in these weird shapes pushing on tissues. So they cause an injury, the immune system comes in, and so the immune system itself, in all these cases, contributes to the disease in many ways. So for example, in a disease like Alzheimer's or neurodegeneration type diseases where neurons might be degenerating, if the inflammatory response comes in, it could contribute to the degeneration of those neurons. It could also protect you from degeneration. It just depends on the situation. But it definitely is a contributor to many of these diseases. Okay, so now we'll turn to how it does it. And before I do, I want to see, is there a clock here? I don't actually have a watch. 6:30. Let me get my something so I can tell what time it is. Not be good if I went too much over. Okay. So what I want to discuss is a little bit of the mechanics of it. How does the immune system do what I showed you in the first few movies? What are the cells that are involved? How do they do it? How do they work? So again, reminding you, their goal is to get rid of the bacteria, the fungi, the viruses, the parasites, the worms. And it has to deal with pathogens that come from many sources. HIV, which comes from people, cholera, which comes from water and food, Lyme disease, which comes from the deer tick, malaria, which comes from the mosquito. So this is a really diverse set of pathogens in terms of their structure, but also in terms of their route of entry. They may come in from any tissue in your body. Okay, so who does the work and how do they do it? So I like to think of the immune system as working through cells, that is direct action of cells on pathogens in some way, or indirect, or through things that the cells secrete, like proteins, and I'll tell you about that in a moment. So example of direct is the following. The neutrophil that I showed you in the first movie. It comes, it sees the pathogen, it gobbles it up and destroys it. Or it puts toxins all over it and destroys it. So macrophages or neutrophils are called phagocytic cells, and phagocytosis means eating. So these phagocytic cells are direct killers. They just go right to the pathogen and eat it and kill it. Now, viruses are a very strange kind of pathogen, and certain bacteria as well. They live inside our cells. This is a cell, and this is a bacterium or a virus. It lives inside the cell. Now, that's a little bit of a problem. These macrophages and neutrophils can't find them, obviously, directly. So they have to have some indirect way. And so the immune system evolved a way to detect an infected cell. It can tell that a cell is infected, and it kills your own cell. And so T cells and NK cells, natural killer cells they're called, these are two cell types that detect infected cells and kill them. Now, of course, there's a risk there because you're killing your own cells, and you're probably going to kill a lot of cells. So for example, if your lung, if you start to kill too many cells, it will be hard to breathe. But this is what you need to do to eliminate an intracellular pathogen. There's no choice. Okay, so these are direct and indirect ways in which cells can destroy pathogens. The other way is through proteins and other molecules that are secreted, that is, they're released by cells. And B cells are the classic cells for this. B cells have these proteins called antibodies on their surface, and I'll show you in a moment a picture more about it. When they recognize a pathogen, they get activated, the B cells get activated, and they release these antibodies throughout the body. And so the antibodies are now circulating everywhere in your body, and they surround the pathogen, stick to it, and kill it in many different ways. There's also another kind of protein that gets released into the body called complement proteins. Your liver does a lot of that. And these complement proteins actually make a hole in bacteria. Just make a hole in it. And I'll show you in a moment how that looks. So this is the example of the T cell that kills. Here's a cell and some viruses that are infecting the cell. The virus gets into the cell, and it's sitting in there. And now the cytotoxic T cell, or the natural killer cell, those two, recognize something on the surface that tells it it's infected and kills it. Here's an example of a virally infected cell with a V in it for virus. And these are the T cells surrounding it. They all have recognized that this is a virally infected cell, and they're about to destroy it. If I showed you the picture a minute later, this would be destroyed and opened up completely. Okay, so that's how it recognizes infected cells. Complement proteins are these proteins. There are many of them. They're called C1, 2, 3, 4, 5, 6, 7, 8, 9. And they form this very, very complex structure called a pore. And this is on the wall of the bacteria. And this is a picture of holes made in a cell. Here's a side picture with an electron microscope of the pore that's made. So it's like a dagger. But it's a very well-designed one in the sense that there are all these proteins that come together in a precise way to make a hole. And when that hole occurs, of course, the bacteria will die because all its contents will be poured out. Okay, so this is a general way. And these complement proteins can recognize and distinguish bacteria from us, for example. They don't do this to us very easily. So how do the antibodies work? The antibodies come from B cells. As I said, the antibody protein is on its surface of the B cell. If it recognizes a bacterium, the B cell gets activated and releases the antibodies throughout the body. The antibodies then bind to the bacterium. And one of the things, for example, those antibodies do is they bring the complement system to the bacterium. And then the complement system will make the pore and kill it. Now, antibodies are amazing because we have trillions of these B cells in the body, and each B cell has a slightly different antibody with a shape that's slightly different from the other one, from the next one. And so any bacterium that comes in or any molecule that comes in can be recognized by 1 or 2 or 100 of these different antibodies. And only the B cells that contain the ones that recognize will start dividing and produce these antibodies to destroy the bacteria. So it's a pretty neat system. And one of the even more impressive things about it is if you get vaccinated, immunized, or infected with something once, you get a certain affinity between the antibody and your bacteria. Some measure of affinity, how stuck it is. If you get infected again, the affinity goes up. If you get infected again, the affinity goes up. So the antibody actually changes over time. The sequence of the DNA of the antibody changes as it encounters this pathogen over and over. And each time it changes, it optimizes the binding to the pathogen. Okay, so it's a real-time adaptation, a real-time genetic adaptation. It's actually like evolution. So the DNA actually changes randomly, and if an antibody binds better, that B cell grows better. And if it grows better, it dominates And the other B cells that don't make antibodies that bind well, they die off. Okay, so it's evolution in action, and this occurs every day, every time you see a pathogen. Okay, so this just describes that each T and B cell, T cells as well, has these receptors with slightly different sequences and shape, so that almost any molecule you encounter can be bound by them. When a pathogen enters, the immune system always has some T cells or B cells that can bind it. When the response has been induced, the cells that encountered it remember for 20, 30, 40 years that they saw it. And that's the basis for vaccines. So that B cell that had the exact antibody that fit that pathogen will forever remember that it saw the pathogen. And the next time the pathogen comes in, it activates itself when it sees the pathogen within hours rather than days to weeks. And so you immediately respond very strongly. In addition, it continuously makes a little bit of the antibody in your body to keep you a little bit protected as well. Okay? So that's the basis for what we call memory. First, the system learns what it sees and optimizes the binding, and then it remembers that. And that's the vaccine basis. That's why you need a different vaccine, of course, for every pathogen, so you get that individual memory for every pathogen. So the immune system is a little bit like the emergency system that we have with police that are looking out for different events that are occurring. And these names, TLR, NLR, RLR, are some of the proteins we study in our lab. These are proteins that detect pathogens. So there are TLR proteins that can detect a bacteria. There's a TLR protein that can detect a certain kind of virus. There's an RLR protein that can detect another kind of virus, and so forth. And so these guys are out here on the surface of a cell, monitoring the environment and seeing what's going on. And then when you see the pathogen, they then alert the cell that sees the pathogen, what kind of pathogen it saw. That cell turns on a very special response that's unique for that pathogen. You can't fight a virus with the same approach you fight a bacteria or a fungus. They all need different mechanisms of attack.
Are these the mechanisms that fail to cause autoimmune diseases?
That fail and therefore cause autoimmune disease, you mean?
Yes.
Yeah, so basically, there's evidence in some cases that these systems are turned on inappropriately. So for example, in lupus, one of these TLRs, TLR 9 and 7 in particular, instead of recognizing, what usually they recognize is the DNA or an RNA of a virus. Okay, so viruses have RNA and DNA just like we do. They recognize that virus, turn on the immune response to kill the virus. In lupus, what happens is, and again, for unknown reasons, why exactly, but this is what happens, they recognize your own DNA and RNA as foreign. They think that it is viral RNA and DNA, and they mount an attack against your own body rather than a virus. Okay? So in lupus, these sensors are tricked. Why they're tricked is a much more complicated question, but that's appears to be what happens. So in mouse models of lupus, if you turn off these genes, TLR9 and 7, both of them, the mouse does not get lupus. Okay, so now I want to get in depth into vaccines and a little bit into allergies, but mostly vaccines, because I think it illustrates a lot. So Jenner, in the late 1700s, noticed, and many other people had noticed it before, that women who were milking cows were somehow less susceptible to smallpox. And I guess it probably wasn't that hard to notice because smallpox is very easy to notice. And it's probably easy to notice when a whole group of people don't seem to have it. And so many people had observed that. And so he thought, well, this is interesting. Maybe there's something in the cow. And so he was able to actually get these cowpox, which is equivalent of smallpox, but in cows, which doesn't cause us much of a sickness, just a mild smallpox. He was able to observe that people get this, and was able to take the little pox on the skin and use those to immunize other people, and then see that they don't get smallpox. Okay, so he was able to prove for the first time that you can immunize in a systematic way. Now, Other civilizations in other places had probably noticed it as well and had used it to vaccinate. But he was at least the first to write something about it, so whatever that means. And then 100 years later, Pasteur is actually the one who figured out what to do about this. This was a very chance observation, okay? Lucky break that there was this other virus that a cow had that happened to immunize against this deadly disease. But what about all the other diseases? And so it's Pasteur who realized that you can take any pathogen, if you can isolate it, and inactivate it in some way, and then give it to a person, and that will immunize the person without giving them the disease. Now, he also happened to see it by mistake. He left some chickens that had chicken cholera to dry, like chicken skin to dry, and left it in the sun for a few weeks. And when he came back, he realized that maybe this material would be interesting to try, and he tried it, and it vaccinated the chickens against this disease. So he realized that if you heat up a pathogen, basically, and that's what's been done for many years, you can inactivate it. But yet it still contains all its components. It's just not alive, right? So all those components are sufficient to vaccinate a person, but not to make a disease. So this began really everything until today. And so inactivating pathogens was just an empirical game that was tried by many, many scientists over 150 years. And so many, many pathogens have successfully been used for vaccination prior to even understanding what an immune system is. So there are a couple of hundred years of work here where none of what we're talking about meant anything, but there was a way to inactivate a pathogen and put it back into a person. So again, the way that worked is the infection created a response, but there was a memory of that response, and when you got infected again, you were protected. And that's the level of understanding they had. Now, in developing vaccines, what was realized, of course, quite quickly is that they have to be extremely effective. Okay? If they only prevent disease in 30% of people, it's still going to spread. The disease is still going to spread. So you need to protect at least 80% to 90% of people, depending on the disease, to prevent the transmission of the disease across the population. So of course, that took a lot of empirical work to get to that level. They also had to be long-lasting. Again, otherwise you have to keep immunizing people. And most importantly, they had to be extremely safe. From the first day that vaccines were put into people, there were entire, very large groups of people who were against vaccines because there were occasionally side effects. Of course, especially a few hundred years ago, where they really didn't know what they were doing. Occasionally, they would get the infection itself if they didn't inactivate the pathogen well enough. So, so the safety is a factor that's today in vaccines considered absolutely essential. And this is just a list of some of the vaccines that have been developed over the years, quite a lot. Huge number of diseases, all the way here from typhoid and cholera to most recently Lyme disease, and this, and here from smallpox to rotavirus. And these are live vaccines versus non-live vaccines. The live ones are attenuated in some way, like in the ways that I told you. They might have a little bit of ability to replicate, but not much. And then the non-living ones are completely inactivated organisms or even pieces of the organisms that are sufficient to protect you, enough to get the immune response to have a good memory of the right thing. And again, the impact of vaccination is this incredible decline, as you can see here in smallpox, as I showed before, also in measles, mumps, and rubella. And you can look at this for any vaccination and see this kind of curve. And, oh, something happened to this slide. So polio is another nasty disease that causes paralysis. And you can see after polio vaccination, there was a constant decrease over many years in polio. And the question is, what happens if you don't get vaccinated? And so there was a group in the 1990s in the Netherlands who refused to be vaccinated for many years. And there was an outbreak. And the outbreak had 71 patients who got infected, 2 died, and 59 had paralysis. So that just reminds you how bad this disease is, if 59 out of 72 people get paralyzed. And so this outbreak was a clear proof that if you don't get vaccinated, you will get it. Now, the thing is that the communities around them, no one got it because people were vaccinated well enough so that it didn't spread at all. Okay? So it only spread within these communities. Okay, so what are the challenges right now in terms of vaccines? The challenges are really, one of the important ones is to develop a test to model vaccines. That is, it's pretty hard to test a vaccine in 100,000 people, but that's what has to be done today. So if we could make models that are better mimics of what the human immune system is like, we might be able to better develop the vaccines and pick between them before we give them to people. And that's, I think, a very important direction. We have to figure out a systematic way to know what to immunize with, which part of the pathogen and why. It's all been empirical so far. How do you figure out what you really need to be injecting, which part? We have to learn more about which immunological responses are the key parts of the protective immunity. For every pathogen, it's a little different, and it will take a lot of time, I think, and years to figure that out. This is not as important, but would be nice to devise needle-free delivery systems, and there are many, many companies and groups devising these approaches. Preparing vaccines do not require refrigeration. That's obviously critical for most of the developing world. And creating ones that can be given at single dose so that you're pretty much done the moment you get the vaccines at early childhood.
Are there vaccines associated with increases in autoimmune diseases or screwing up the sensors years ahead?
Not that we know of at this point. So, I mean, I think that there have been epidemiological studies that have tried to look at that, and there doesn't seem to be a very strong correlation in any vaccine. But I think these are, large-scale studies that take a long time to do, just like any epidemiological studies. So if there's a very small fraction of people, it's going to be hard to find them. Okay, so back to allergies. So allergies can be caused by drugs like penicillin. People are allergic to that. By venoms and by peanuts, by animals, of course by pollen, dust mites, cat danders, shellfish, milk, eggs, fish, an enormous amount. And it can be through different routes of entry and lead to very different levels of responses, all the way from being unable to breathe to having diarrhea and vomiting. So a very complex set of responses. So what is allergy? Why do you get it, and how does it work? So the answer to the first question, why, is we still don't know exactly why we get allergies. But we know a lot about how. So what happens is, here's a B cell again. Here's the antibody on its surface. And I mentioned antibodies can recognize anything. Okay, so what's the risk of recognizing anything? It means that it might recognize something benign, something that really doesn't cause any problem. So pollen is really not hurting anyone. But when it comes into the body, there is a B cell that recognizes that pollen. Now, the pollen is recognized. It stimulates that B cell because the B cell is like, oh, I found something. The B cell gets activated. It produces large amounts of the antibody. It's a little hard to see. Those antibodies then circulate throughout the body. And one of the things they bind to is something called the antibody receptor, or the IgE receptor, on a mast cell. And I'll tell you about mast cells in a second. But mast cells basically cause all the symptoms that you feel when you have allergies. The next time you see that allergen again, those IgE antibodies are all throughout your body. The moment the pollen comes in, it binds to it, and a bound IgE antibody will stimulate the mast cell and cause all the symptoms that you recognize. So the first time you see it, it's not too bad. But the second time, and especially as this IgE level, the antibody level, keeps increasing in your body, when it's at very high levels, the moment it interacts with that allergen, whatever that allergen is, whether it's from pollen or dust mite, whatever it is, it will bind to the mast cell and activate it. And when the mast cell is activated, it releases, there are these little vesicles called granules that release a lot of nasty toxic things. And they cause all kinds of problems like airway congestion and causing you to want to empty your gastrointestinal tract. I'll tell you why in a moment. And increasing the blood flow. Now, why is this? What are these mast cells for? They're obviously not to get rid of pollen. This is not something you should be doing to get rid of pollen. So it ends up that the main role of the allergic response in the body is not one that we see in the United States, but in the rest of the world you see it every day. And that's to protect you from worms. So what the mast cells, what the IgE antibodies do, is they recognize worms, they surround the worm, and then the IgE antibody activates the mast cell. The mast cell binds all around the worm, and another cell called the eosinophil, which has some similarities to the mast cell. They surround the worm, and they release all these toxic compounds. It's pretty hard to kill a large organism, right? So you need a massive amount of material that will kill it. And of course, you want to be able to have diarrhea to remove that worm if it's in your gut and so forth. So there are a lot of systems in your body that are modified in order to eliminate the worm, and there are a lot of toxins that basically will cause tissue damage, but also would kill the worm. So one of the theories for why we have allergies here is that we don't have worms. And so our immune system doesn't have much energy put toward the worms, and it puts it toward allergens. Yeah?
Get some of the more complex reactions, such as oxidative necrolysis, when the body essentially sheds its skin, you actually see cause for it.
When it sheds its skin, is that what you said?
Yeah, how it's something that.
Why is that related to worms or something?
What?
What's the question?
Why would something like that happen?
Yeah, so when we have all these reactions where tissue damage occurs or the blood flow increases in a certain area and you get swelling and all that, that's part of the normal immune response. The immune response includes attacking a site, right? So if you have a pathogen there, you're essentially attacking the site. So the immune system will make a mistake and attack many different sites, including the airways, the skin. It depends on the allergen, the disease, the route of entry, and that can affect different tissues in your body in this inappropriate way that normally would probably be getting rid of something that is actually there. Yeah, that's probably doesn't happen. So I think the allergens give you even a stronger response that's more broad in some cases. Okay, so what are the challenges for the future? The big challenges I mentioned already: HIV, malaria, TB, HCV. HIV, malaria, and TB, if they were eliminated through vaccines, would single-handedly change the economics and growth of Africa because they cause a huge amount of disease burden that blocks people from doing anything in their life, taking care of their family, working. So this is a very major priority, and the Gates Foundation is a good example of a recent, a lot of energy and money put into these problems, which is fantastic. Autoimmunity is, really only appreciated in terms of the mechanism in the last 20 years. So it takes a long time from starting to understand something to making a therapy. And one of the first ones is for arthritis, Remicade, Enbrel, and these other antibodies that are actually given to you. And the antibodies are actually antibodies that block the immune response. So they actually bind to an immune molecule and block it from working. So the antibody is being used against the immune response, which is not normally how it works, but that's by design. So there are cases where there are some more sophisticated treatments. But this one was based on a concept that came 20 years ago and is really not even close to up to date on what we know about autoimmunity now. So a lot more is being done in terms of mechanisms, but it will take a lot of years, I think, to understand autoimmunity well enough to develop these things. Allergy is still not, we don't know how to cure it. I mean, there's desensitization where you get the allergen in small amounts, and it tends to turn off the immune response in a certain way that is slightly understood. There are, of course, many inhibitors that you take, antihistamines and others, which can relieve you of symptoms and make it livable. But we don't have any systematic way to remove allergy from occurring. And then the last one I want to speak about is cancer. And there, I think, is very exciting because cancer is a very hard disease to treat, hard disease to make drugs for, because a few reasons. One is every cancer is different in your body, okay? So it's very hard to make a drug for every cancer. That's a lot of work. The second is in some really nice cases in the last year where a drug was made specifically for one cancer, like melanoma. There's a protein called BRAF, which is mutant in melanoma and causes melanoma. And a drug was developed and was given in clinical trials recently. And immediately the melanoma disappeared. But a few weeks later, it started to come back up. Now, why is that? And went right back to normal on its normal course. The reason is that tumors are able to mutate their DNA more readily than your normal cell. And so the DNA that encodes for the BRAF protein became mutated, and some melanoma cells had a mutation that didn't allow that drug to block that protein anymore because the shape of the protein changed, so the drug couldn't recognize it anymore. Right? The drug couldn't bind. The cell continued to grow as if it never had seen the drug. The melanoma grew back up. And so it's very difficult to develop drugs to destroy tumors. And chemotherapeutics and radiation, of course, are what we use most of the time. And those are just not specific. And some diseases, the diseases are sensitive enough that it works to some level. And in most diseases, it only slows down a little bit. And in many diseases, it doesn't do anything. But it's the only thing we can do. So one of the exciting directions, I think, in immunology is to develop immunotherapies and vaccines for cancer. And this is a hard problem, but there are a lot of people who are interested in trying to do it because cancer is a leading cause of death in this country. So the only pure success story, and for a very simple reason, which is cervical cancer, where human papillomavirus is the cause of most female cervical cancer. It happened to be that this virus actually causes cancer. Probably most cancers are not due to viruses. There are some cancers that are. For those that are, since we're pretty good at making vaccines against viruses, it worked out pretty well. And so there are about 500,000 new cases a year with 250,000 deaths per year. And the vaccine was given here to 10,000 people, or a placebo, meaning no vaccine. And you can see that the people who got the vaccine, there was 0 to 1 case of the cervical cancer. And in the people who didn't get the vaccine, there was the normal rate, 56, 50, 85 out of the total number. So that prevented the cancer. So this was a very exciting direction for the cancer world, the idea that you might be able to not even get the cancer. But of course, that's only going to work in that way for cancers caused by viruses. For the average cancer, almost all, that is, are not caused by viruses. They're caused by mutations in your own genes. So how are we going to get immunotherapy to kill those? So there are a couple of ways. One is antibodies. So I told you antibodies can be made to recognize anything. And so people have developed antibodies that recognize certain forms of breast cancer that express the HER2 gene, the Herceptin gene, on their surface. And that antibody can bind to those tumors and help eliminate them through immune responses. And so that's one way, is with antibodies. The other way is to convince T cells to kill them. Okay, so I mentioned earlier that cytotoxic T cells can kill cells that are infected with a virus. What if you can convince the cytotoxic T cells that there's a virus in a cancer cell, even though there isn't? And then the T cells will find those tumors and kill them. Those are the two ways that people are working on. And two recent examples. One is with an antibody that actually does something very unusual. It blocks something that blocks the immune response against tumors in your body. And when that was done, this is a survival curve. You can see the upper curve is with this anti-CTLA-4 antibody. People actually lived longer. More people lived longer in melanoma. Melanoma, every drug that had been given pretty much did not increase survival in melanoma. So this is the first one, really, that increased the survival time. And there's a second drug as well now called anti-PD-L1 that's working even better than that. So I think that there's at least what we call a signal. At least we can see something happening with this. The other one is a melanoma vaccine given together with this anti-CTLA-4. And this is just an example of a metastasis of the melanoma that was reduced after vaccination. And you can see in aggregate in these patients, there was a reduction in the size of the tumor in these patients. And this is all with immunological therapy. You're not directly killing the tumor through a drug that kills the tumor. You're simply turning on an immune response that then kills the tumor for you. This particular graph is from my wife's lab. She's an oncologist at the Dana-Farber who makes vaccines to tumors. And this is for chronic lymphocytic leukemia. And here she gave this patient vaccines, 6 doses against their own tumor. And after several months, the amount of tumor went below the detection level that we can detect tumors. And so there are examples now that are developing across the world with different tumors of examples of success. So the challenges for us, and this is something that my wife works on a lot, and I'm also helping her now as well, is what do you immunize with? What part of the tumor is your thing that you're immunizing with? Just as we talked about vaccines, what's the equivalent of an inactivated pathogen? So the most common thing that's done is actually an inactivated tumor. The tumor cell is killed and then injected as a dead cell together with a stimulus that turns on the immune response. And that's the way the ones I showed you before worked. Okay? So it's exactly the equivalent of the same vaccine we've given for pathogens for hundreds of years. The problem with it is that most of the pieces of the tumor look exactly like every other part of your body. So in theory, if you give too strong a vaccine, your immune response will mount against the rest of your body and will cause autoimmunity. And so that's a case where autoimmunity can develop in a vaccine because you're immunizing with yourself. Because a tumor is 99.9999% identical to the other cells in its neighborhood. It just has a few little changes that are unique to the tumor. So a question is, what do you immunize with? And so a strategy that we're interested in taking is to immunize with just the parts of the tumor that are different from everything else and ignore the rest of the tumor. And that's the approach we're taking now. The second question is how to induce a strong immune response. Immunity to self is not something your body does very well. It has all these mechanisms to prevent immunity to self, so you don't get autoimmunity. Most people don't have autoimmunity. Why don't they have autoimmunity? Because the tissues, the cells of the body have mechanisms, have methods to stop the immune response from attacking it. So you have to overcome that in the case of a tumor vaccine. You have to convince the body to make a response. And so the antibody I showed you before, anti-CTLA-4, is the first invention of a drug that will actually increase the immune response against self. So when combined with the correct thing to immunize against, we believe that that will give you a strong immune response against the tumor, but not the rest of the body. And so that's essentially the direction we're going. OK, so I think it's probably time to finish, but I'll just mention one last thing, which is what we do at the Broad. So at the Broad Institute, it's really a post-genome institute. That is, it used to be a genome institute. The genome was sequenced here and sequenced across a few other centers in the world. But now we're in a post-genome institute because the genome has been sequenced. So what do we do after the sequencing of the genome? And so now we have this problem, which is we've got this very complex circuit, and things are happening, and we want to be able to measure things in it, perturb it, and see how it works. And so any cell can be looked at that way. And the cell has, as we know, 23 chromosomes. Chromosomes are translated into proteins and so forth. And the question is, what is the role of this is pretty light. What's the role of the 20,000 genes in your body in human health and disease? That's our goal right now. And if we can get there, we can get more effective therapeutics, disease-specific therapeutics, individual-based therapeutics. And how are we going to get there? And the idea we have, really, is that we need to determine the role of each gene. So let's say each switch here is a different gene in the body. We want to know if you turn off a switch or turn it on, what happens to the body? If you turn off a gene and you monitor all the changes in cells or in the body, what happens as a result? Or if there are mutations in the gene naturally in people, what disease or human trait is that associated with? Those are really the questions that the Broad Institute was built to figure out. What do all the genes do in disease and in health, in cells? And then, if you can figure that out systematically, you pick the ones you think are the ones you want to move around a little bit, shift, make them a little stronger, a little weaker for different diseases to try to re-equilibrate diseases, when possible. And so this is an example of a diagram that generated in our lab of a circuit of a cell. And these are all the proteins, or many of the proteins, not all, in a cell that are involved in responding to the influenza virus. So we were able to find them through a systematic search. We turned off every one of these genes individually in the cell and asked, how does that affect the response to influenza? How does that affect the infection by influenza? And we were able to say, these are the genes of the 1,000 others that we also looked at, which are not shown here. These are the ones that actually affected influenza. Okay, so we're able to reduce this complex web of interactions and proteins down to the ones that are relevant for this process. So this is what we need to do now in a systematic way, and what we do here. Okay, so thank you for coming, and thank you for Megan for organizing these talks, and for my lab and department for everything. So I'll take questions.
My question is about pathogen scorecard.
Yeah.
A century ago, would it have been different?
Well, TB has always been infected billions of people. So that is actually very, very true. But in terms of what would be killing you the most, it might be cholera or smallpox, other diseases that are basically almost eliminated now, but were better killers. TB, although 2 billion people are infected, only several million die per year. But I showed you the example, for example, with smallpox or polio, where the percentage of people who die or get paralysis is much, much higher. So some of the diseases we had earlier were much more damaging. So the scorecard would have been different in terms of disease. But in terms of, would those still be up there? Yes. But maybe they'd be less in terms of how much damage they cause. Yeah.
Some of these viruses are more clever than some other viruses.
Right.
So it seems as though once you developed a vaccine for polio and smallpox and so on, that worked. But the common cold and the flu and TB and some of these are really smart. Do you have any idea what causes this dysfunction?
Right, so the question was, why are some viruses so clever that we can't make a vaccine against them, whereas others we've been able to for years and years and years? Without knowing anything, we were able to do it. So the common cold is a little more complicated because there are just so many strains of virus around. They're not clever, actually, but they don't do too much to us. But there are just so many of them that it's just very hard to make enough vaccines. HIV is, unfortunately, has this very strange property that it eliminates your T cells. Okay, so a person that has HIV that develop AIDS, their T cell count goes down, 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 down. So at a certain point, if your T cell count goes too low, it's just not effective immunity. There's nothing you can do about it. So HIV would be that way. Influenza simply changes. The vaccine does work. It's just that occasionally it changes because it's in pigs, and chickens and ducks and geese. And so it keeps incubating there and mutating independently of us. And then someone sees a duck and gets that infection. And that starts a whole new process that we hadn't seen before. So that's just bad luck that this is a pan-animal infection. So for each virus, the answer is completely different, or for each bacteria, why we can't do it. TB is another one where it completely seems to inhibit the immune system. You can have a strong immune response, and the TB just sits there and blocks the macrophages from killing it. It has like 10 tricks, which we don't understand, that block it. And so it's an interesting problem, but we don't know how to fix it.
Can you talk about lifestyle causes of lymphoma?
Lymphoma? So lifestyle causes of lymphoma. I don't know much about lifestyle causes of lymphoma. The traditional information about that, the only thing that was known many years ago was that exposure to radiation, because they were in nuclear bombs or in nuclear reactors, will cause lymphoma. But that is not the reason why people in the United States get it. I don't think there is any particular cause that we know of. No, epidemiologically, I haven't heard of any specific cause of it. There are some lymphomas that are due to viruses, but that's not most of them. So I don't think there's anything one can change in one's lifestyle for that. Yeah.
You said that you were able to take those genes and turn them off on
So the question was, is it good enough to turn off one gene at a time, or do you actually need to turn off two at a time sometimes to understand what they're doing? And the answer is you absolutely have to turn off 2 or 3 at a time to understand the network, because these genes work, the proteins work together to do things. And you get rid of one, and you don't really understand how they work together. You have to get rid of both to eliminate some process from occurring, because they may be redundant with each other. We just are very primitive. This is all we can do. We can do 2 sometimes.
The relationship between the immune system and one's emotional or mental state.
Right, so the question is there a relationship between emotional state and immune system? And there is. Stress and all the hormones that are produced when you are stressed in a kind of fight or flight reaction are undoubtedly inhibitory to the immune system. There's enough evidence, mechanism to support that. So depression itself even can probably cause some problems. So a lot of different emotional states can change it. I won't say it changes it radically. It changes the activation state a little bit, but maybe enough in some cases to increase your chance of infection. Yeah.
What would be the cause of reactions towards, say, blood cells from organ transplants, or say black drug, and cancer that's going around
Yeah. So the question is, why do we reject transplants? Is that the first question? So transplants we reject for very strange reasons that would take probably a few minutes to explain. But essentially, our T cells don't recognize them as your own cells, because T cells are trained when they're developing to only recognize your own cells. It's a very strange process that teaches them that. And when they see a cell that isn't yours, they recognize that certain molecules on their surface are different, and they attack them as if they're not self. So that's the reason why transplant rejection occurs and why you need to match people. It's called matching. So you need these proteins called HLA proteins to be identical pretty much between people. If they're not identical, the T cells recognize them as foreign. And then you were asking about cancers that are transmittable. There's only one that I know of, the Tasmanian devil, which is this strange animal where there's a cancer that actually spreads through the population. Why don't they reject their cancer? Don't reject. Could be. Yeah.
What is actually happening in the different types of lymphoma?
In which ones?
In lymphoma, what's actually happening in the body?
Oh, what's happening in lymphoma? So in lymphoma, it is a disease of immune cells, but it's not an immune disease. It just happens to be that these immune cells are dividing or not dying. So they're dividing too much. So as I showed you before, when you have an immune response, the T cells or the B cells divide like crazy, right? But then when they're done with the immune response, they actually die at the same speed. So the curve goes up and then goes down. So what happens in lymphomas is the T cells and the B cells simply do not die. And so one of the mechanisms that kills those cells, that internal mechanisms that destroy cells when they divide, isn't functional. So you get mutations in those genes, and the T cells and the B cells simply stay forever. And so in some lymphomas, it's a very slow process, like in chronic lymphocytic leukemia. The T cells and the B cells grow very slowly over time. And in others, acute lymphomas, they grow very quickly, just like an immune response. So it's basically lack of control of the elimination of the immune response to this.
And then what happens? What's the result?
The result is just that your blood is filled with immune cells. And so at some point, they metastasize, because there's so many of them, that they start to break open tissues and metastasize to other places.
So I'm going to take a pause in question and answer. I'm sure that Nir would be more than happy to answer additional questions if anyone wants to come down. But for now, I want to invite you. We are having a reception in the lobby if you'd like to join us. And also there will be 3 more lectures this season for our series, all of the Wednesdays in July in this room at 6 p.m. And before we all adjourn to the reception, let's give Nir one big round of applause. Thank you.
Thanks.